Hello everyone, I am Dr. Deepika Malik and today's topic of discussion is immobilization of enzymes and their applications. Enzymes are protein molecules produced by living cells which work as biological catalysts and speed up chemical reactions in living organisms. They have the ability to catalyze reactions under very mild conditions with high degree of substrate specificity, thus decreasing the formation of byproducts. Enzymes can catalyze reactions in different states as individual molecules in solution and as attached to surfaces or immobilized. Now let us see the definition of immobilization and immobilized enzymes. Generally, enzymes are found in free form, hence they are not stable and cannot be recovered for reuse. Immobilization of enzyme refers to the technique anchoring the enzyme on an inert or chemically non-reactive surface and insoluble support for their stability and functional reuse. By employing this technique, enzymes become more efficient. It allows enzyme to be held in place during reactions. Attached or immobilized state has been of particular interest. Immobilized enzyme refer to enzymes physically confined or localized in a certain defined region of space with retention of their catalytic activities and which can be used repeatedly and continuously. The first industrial use of immobilized enzyme was done in 1967 by Chibata and co-workers who developed the immobilization of Aspergillus oryzae amino acylase for the resolution of synthetic racemic D and L amino acids. In some industrial processes, whole microbial cells containing the desired enzyme are immobilized and used as catalyst. This technology also prevents the loss of enzymes into the reaction medium, that is leaking of enzyme. Now let us discuss the characteristics of immobilized enzymes. They may be attached with a carrier. They should have great stability. They should show high activity than free enzymes. They should be cost effective. They should be held in place during reactions. They should be temperature and pH resistant. They should be recovered for reuse after reactions for lowering the total production cost of enzyme mediated reactions. They should have protection against degradation and deactivation. They should have the ability to stop the reaction rapidly by removing the enzyme from the reaction solution. They should be easily separated from the product, and product should not be contaminated with the enzyme. Immobilized enzymes shows various applications in different fields. They are used in industrial production of antibiotics, beverages, amino acids, etc. For biomedical applications, immobilized enzymes are widely used in the diagnosis and treatment of many diseases. Immobilization techniques are effectively used in drug delivery systems, especially to oncogenic sites. In food industry, enzymes like pectinases and cellulases are immobilized on suitable carriers successfully and are used in the production of jams, jellies and syrups from vegetable and fruits. Immobilized enzymes are also used for the production of biodiesel from vegetable oils. In wastewater management, treatment of sewage and industrial effluents are also done by immobilized enzymes. In textile industry, scoring, biopolishing and desizing of fabrics are also done using these enzymes. In detergent industry, immobilization of lipase enzyme for effective dirt removal from clothes has also been implemented. Now let us see characteristics of an ideal carrier matrix for enzyme immobilization. The matrix or support immobilizes the enzyme by holding it permanently or temporarily for a brief period of time. There are a wide variety of matrices or carriers or supports available for immobilization. The characteristics of the matrix are paramount in determining the effectiveness of the immobilized enzyme system. They should be inert or non-reactive. They should be physically strong and stable. They should be cost-effective, easily available, regenerable, and should show reduction in product inhibition. Now let us see the classification of carriers. Carriers can be classified into three main categories. First is inorganic carriers, second is organic natural carriers, and third are synthetic carriers. Inorganic carriers have high pressure stability and may undergo abrasion. Examples include commercially silicon dioxide materials like porous glass, silica, zeolites or diatomaceous earth. Mineral materials or clays like silicate, syntony. then ceramics, activated carbon, and charcoal. The second category of carriers, that is organic natural carriers, are favorably compatible with proteins. Examples include dextrin, cellulose derivatives like diethylaminoethyl cellulose, that is DEAE cellulose, or CM cellulose, polysaccharides like agarose, alginate, chitosan and chitin, collagen, carrageenan, gelatin, cellulose, starch, and pectin. And last, synthetic carriers have high chemical and mechanical stability. Examples include polystyrene, polyvinyl acetate, polyvinyl chloride or PVC, acrylic polymers, or polyethylene glycol. Now let us study different methods for enzyme immobilization. Ways of immobilizing enzymes falls in two broad categories. First is reversible or physical enzyme immobilization methods, and second is irreversible or chemical enzyme immobilization methods. Now we'll study both the categories one by one. Now coming to the first category, that is reversible or physical enzyme immobilization. This method is reversible and the enzymes can be removed from the support easily by simple reactions or reversal of the conditions by which the immobilization was carried out. Physical methods are characterized by weaker, non-covalent interactions such as hydrogen bonds, hydrophobic interactions, van der Waals forces, affinity binding, ionic binding of the enzyme with the support material, or mechanical containment of enzyme within the support. This is because of economic reasons, as the cost of the support is often a primary factor in the overall cost of immobilized catalysts. The methods can be of the following types: adsorption, chelation, formation of disulfide bonds, ionic binding, affinity binding, entrapment, and encapsulation. Now we'll discuss all these types one by one in detail. First type is adsorption. It involves the non-covalent physical binding of the enzyme on the surface of carrier matrix or an inert support, which may be inorganic or organic. The process of adsorption involves the weak interactions like van der Waals or hydrogen bonds. The absorbed enzyme can be easily removed by minor changes in pH and temperature. Carriers used for adsorption are alumina, silica, bentonite, cellulose, cephadex, agarose, collagen, charcoal, etc. An example of enzymes immobilized by adsorption includes catalase and invertase. There are certain advantages and disadvantages of the process. Advantages includes that it is a simple and economical process. There is a limited loss of activity as the native structure of the enzyme is less disturbed. They can be recycled, regenerated, and reused. Disadvantages includes relatively low surface area for binding, therefore temporary immobilization is provided by it. Exposure of enzyme to microbial attack is more by this method of immobilization. Yield are often low due to inactivation and desorption. Second type is chelation. Chelation or metal binding is another common type of reversible enzyme immobilization. Being reasonably expensive and involving safety issues, this method is less popular in industries. It is based on the ability of charged and polar amino acids like histidine, lysine, phenylalanine, cysteine, and tyrosine to bind to metal ions via coordinate bonds. The metal ions bound to the carrier surface have metal ligands weakly bound to them. Upon exposure of the enzyme to the carrier, the weak ligands are replaced by the enzyme molecules. The binding can be easily reversed either by introduction of a ligand with greater affinity for the metal ion, like ethylene diamine tetraacetate or EDTA, or by addition of excess of a competing ligand. Carriers used for chelation immobilization are mostly organic materials, usually cellulose, chitin, or silica-based carriers. They are activated as the enzymes bind to the nucleophilic groups by coordinate bonds. Transition metal salts or hydroxides may also serve as carriers. Now, advantages of binding by chelation is that the binding is reasonably strong, due to which enzyme leakage can be maximally restricted. Easy regeneration of the support without any appreciable effect on the yield is possible using chelation method. Disadvantages includes that the reagents involved may not be safe for the production of food products and may cause health hazards. Reduction in enzyme activity due to interactions with the metal ions at the active sites of the enzyme is also a major drawback of this process. Third type includes the formation of disulfide bonding. This technology involves the formation of disulfide bonds between the enzyme and the matrix. Though it is a form of covalent bonding and irreversible enzyme immobilization method, it is classified as a reversible technique because of the ease of reversal of the binding. The enzyme immobilization step requires formation of disulfide linkage between the carrier and a free thiol group, usually on cysteine residues. The disulfide bond is reasonably stable, especially under physiological conditions at which many enzymes function. The binding can be reversed by the addition of reagents such as dithiotritol DTT under mild conditions or by altering the pH. Carriers used for disulfide bonding are generally inert substances like silica, which are chemically activated by agents such as iodoacetate or melamide. Now, the advantages of the disulfide bonding is that proper maintenance of pH and temperature can restrict enzyme leakage as the disulfide bonds are sufficiently stable. The activity of the thiol group can also be altered with pH. Disadvantages is that in a reaction mixture, the pH and substrate concentration constantly changes as the reaction progresses. These may alter the enzyme binding, consequently leading to enzyme loss. Another type includes the ionic binding. This is a simple reversible mode of immobilization of proteins which involves ionic interaction between the enzyme and the support. The support used is generally charged such that the protein to be bound has an opposite charge. The enzyme is therefore bound to the support via ionic interactions. It can be easily reversed by altering the pH or salting out of the enzyme. Advantage is that it is very easy, inexpensive and requires simple inputs for reversal of the binding. To maintain an optimum pH during their action tenure, easy manipulation of the acidity or alkalinity in their action mixture can be performed as the matrix which immobilizes the enzyme is stably charged. Disadvantage is that the presence of the charged support causes several problems like enzyme structure distortion and alteration in enzyme kinetics. High charge has the potential to disrupt the enzyme catalysis. As a result, maximum yield is hindered. Fifth type includes the affinity binding. The technique is based on high affinity interaction between biomolecules or the antigen-antibody interaction. The carrier matrix is synthesized specifically for a single type of enzyme and contains antibodies against specific epitopes of the antigen or enzyme. Advantage of affinity binding is that the reaction is highly specific and no containments are present on the carrier. If the antibody on the support is highly specific for the enzyme, the step of the enzyme purification can be bypassed. Enzyme from an impure solution can also specifically attach to the matrix. Maximum activity of the enzyme is also ensured if the antibody is targeted at an epitope away from the activity site. Disadvantage is that the method involves use of specific antibodies which are generally very expensive. Next type includes the entrapment method of immobilization. In entrapment, the enzymes or cells are not directly attached to the support surface but simply trapped inside the interstitial spaces of a cross-linked water-insoluble polymer matrix as you can see in the image. Enzymes are held or entrapped within the suitable gels or fibers. It is done in such a way as to retain protein while allowing penetration of substrate. It can be classified into lattice and microcapsule types. First is inclusion in gels like polyacrylamide gel, polyvinyl alcohol gels. Inclusion in fibers like cellulose and polyacrylamide gels. Inclusion in microcapsules like polyamine, polybasic and acid chloride monomers. Advantages of entrapment method is that cost is low, they are industrially useful and can be reused many times. Disadvantages includes that the preparation is difficult, the enzyme may leak from the pores and there is lack of control on the process. Next type includes microcapsule type entrapment or encapsulation or membrane confinement. It involves enclosing the enzyme within semi-permeable polymer membranes. Membrane is made up of nitrocellulose and nylon. In this process, semi-permeable membranes work as base which help to hold enzyme at one place during whole reactions. The membrane should be polymeric and non-ionic in nature. Biopolymers like alginate, maltodextrin, cellulose, chitosan, etc. are available which can be used for the purpose of encapsulation of an enzyme. Advantages includes that there are no chemical modification, relatively stable forms are produced, and easily handling and reuse of the enzyme is possible. Disadvantage is that the enzyme may leak from the pores, cost is high, and they are difficult to prepare. Now, second category of enzyme immobilization includes the irreversible or chemical enzyme immobilization methods. Reversible immobilized enzyme technology cannot fulfill the aim of using an immobilized enzyme on a long-term basis. Therefore, irreversible or chemical enzyme immobilization methods can overcome this limitation. Irreversible immobilization involves strong chemical bonds like ether, thioether, amide or carbamate bonds between the enzyme and support material, and particularly serves to maintain reasonably stability of the enzymes over a long period of time. Most industries use enzymes immobilized by these methods, thereby allowing continuous processing of the substrates without the need of replacing the enzyme very often. There are two main types under this category. First is covalent binding and second is cross-linking. Now let us discuss these two types one by one. Covalent binding is based on the immobilization of the enzyme by the binding of enzyme and water-insoluble carriers by covalent bonds. Cellulose, dextrose, agarose are some of the important support in covalent attachment. The choice of support depends upon the surface properties. The support should have functional group to facilitate the attachment. The functional groups that may take part in the binding are amino group, carboxyl group, sulfhydryl group, hydroxyl group, imidazole group, phenolic group, thiol group, etc. The advantages of covalent binding is that the binding force between enzyme and carrier is so strong that no leakage of the enzymes occurs, even in the presence of substrate or solution of high ionic strength. Also, enzyme activity is high. Disadvantages includes that covalent binding may alter the conformational structure and active center of the enzyme, resulting in major loss of activity or change of the substrate. Regeneration of enzyme is possible by this method. Second type of irreversible immobilization technique includes cross-linking. It is different from other techniques in the sense that it does not require a support for the immobilization. For most enzymes, the cross-linking agent used is glutaldehyde, which is cheap, stable, and easily available. However, glutaldehyde partially or totally inactivates some enzymes. For biomolecules, alternative cross-linking agents such as dextrin polysaccharide, bis-isocyanate, bis-diazobenzidine, diazonium salts, and functionally inert proteins such as bovine serum albumin or BSA should be preferred. The advantage of cross-linking is that the enzyme is strongly bound. Higher stability, that is pH, ionic and substrate concentration, is also shown by the enzymes immobilized by this method. Disadvantage includes that cross-linking may cause significant changes in the active site of the enzyme, and not a cost-effective process it is. Now let us see the advantages of using immobilized enzymes in industrial processes. There is an increased functional efficiency of enzyme. Enhanced reproducibility of the process they are undertaking is also shown. There is a continuous use of enzyme. Less labor input in the processes is required. Saving in capital cost and investment of the process. There is a minimum reaction time. Less chance of contamination in products. More stability of products obtained. Suitable for industrial and medical use. Enzymes are held in place during reactions. They are temperature and pH resistant. They are easy to be recovered for reuse after reactions, and enzymes become more efficient. Now there are certain limitations also involved with enzyme immobilization. These are cost of carriers and immobilization, changes in properties of the enzyme, mass transfer limitation, problems with cofactor and regeneration, problems with multi-enzyme system. Activity loss during immobilization. Industrial applications are limited and only few industries are using immobilized enzymes or immobilized whole cells. Enzymes are inactivated by heat generated in the system. Now let us have a quick review of the topic. The next slide shows diagrammatic representation of each type of immobilization. First category of immobilization technique includes reversible or physical enzyme immobilization methods. First is adsorption, where there is covalent physical binding of the enzyme on the surface of the carrier matrix. The process of absorption involves the weak interactions like van der Waals or hydrogen bonds. Second type is chelation, where the charged and polar amino acid of the enzyme is bind to metal ions, which are further bound to the carrier surface. Third type is the formation of disulfide bonding between the carrier and a free thiol group of cysteine residues of the enzyme. Fourth type is the ionic binding, where the protein which is charged is bound to a charged carrier of opposite charge. Fifth type includes the affinity binding, where the carrier matrix is synthesized specifically for a single type of enzyme. That is, the carrier is given a shape specific for the desired enzyme to fit in. Sixth method includes the entrapment, where the enzyme is not directly attached to a support surface but are simply trapped inside the interstitial spaces of a cross-linked water-insoluble polymer matrix. And the seventh type includes encapsulation, where the enzyme is enclosed within semi-permeable polymer membranes. Second category of immobilization technique includes irreversible or chemical enzyme immobilization methods. In first type, there is a covalent binding between the enzyme and water-insoluble carriers. Whereas in second type, that is cross-linking, a cross-linking agent is used like glutaraldehyde, which holds the enzyme very tightly in place. With this, we wind up the topic of immobilization of enzymes and their applications. For any doubts and queries, you can contact me through the given email ID. Thank you.